Sri Gurubhyo Namaha. Welcome to our online teaching academy Ekalaiva, and I welcome you all for the biology class. This is class 11 biology. Last class we completed about the central nervous system. And now we are going to talk about the other types of nervous system. So what are the different types of nervous system? Central nervous system, the other one peripheral nervous system. In that the divisions also we are going to talk about. PNS, it's an abbreviated form which is called as peripheral nervous system, is a division of the nervous system and it contains nerves. It lies outside the central nervous system. This is the location of PNS, which lies outside the CNS. And then one by one we will see about the role also. So what does the peripheral nervous system do? Number one, it connects the central nervous system to the organs like limbs and skin. It carries sensory and motor information to and from the central nervous system. 3. It allows the brain and spinal cord to receive and send information to the other areas. 4. It also regulates the involuntary body functions like heartbeat, breathing, etc. So this is about our PNS function, the role. If you see the flow chart, PNS, otherwise called as peripheral nervous system, divided into sensory and motor. In motor we have autonomous, in autonomous we have sympathetic and parasympathetic. We are going to see this role, sympathetic and parasympathetic. Autonomous nervous system, another one somatic nervous system. So we will recap once again. PNS or peripheral nervous system classified into two, sensory, motor. In motor we have autonomous and somatic. Autonomous divided into two, sympathetic and parasympathetic. So what is sympathetic and parasympathetic? They have an antagonistic relation. What is antagonistic? Like opposite. If one is going to control, the other one is going to induce that. So the opposite functions, we call this antagonistic reaction. So here we will talk about sympathetic and parasympathetic. If you see our system on the whole, you can see how it has been divided. Okay? One side parasympathetic, another side sympathetic. For example, eye, pupil if you take, this sympathetic nerve will dilate and here it constricts. Another example, inhibits saliva production, which one? Sympathetic. And here it stimulates. So one is stimulating, other one is inhibiting that. So it has to be in a regulated way. So this system controls on the whole, sympathetic and parasympathetic. You can see here the coordination, how it coordinates and works together, okay, which is under PNS, peripheral nervous system. Next we will talk about the spinal cord, which is a part of CNS, central nervous system. Already we completed brain. Now we are talk about the spinal cord. Location, where it is located. Brain if you take, it is located in this case, okay, cranial we can call it as. The next part, spinal cord, from the foramen of magnum, okay, a cord runs towards the back side and that cord protected by a vertebral column. In the locomotion movement lesson, we saw about the vertebral column. We have different parts like cervical, thoracic, lumbar, sacral and coccygeal. You can see the same thing here. Okay, I will repeat, cervical, thoracic, lumbar, coccygeal and sacral. The 5 parts you can see. Okay, thoracic, lumbar, sacral and coccygeal. So these bone case, okay, vertebrae, okay, it is going to protect the spinal cord. Spinal cord is nothing but the bundle of nerve fiber. And depending upon the location, if you see the number, okay, the number of vertebrae you can see. So here it is 1 to 7, cervical, thoracic if you see 1 to 12 in number, lumbar if you see 1 to 5, sacral 1 to 5, coccygeal a tail-like appearance which is a fused one which is 1 in number. Actually it was 5. It's fused together, looks like a tail outgrowth and it's 1 in number now. So what is spinal cord and what its role? Its major role seen in reflex action. When you touch something hard, you withdraw your finger or the hand, it's all because of the reflex, okay, the immediate reflex. So spinal cord is a long, thin tubular structure. It is made up of nervous tissue. Again I am saying it's a bundle of neuron which extends to form medulla oblongata. Sorry, from the medulla oblongata and then the brainstem. Okay. So, the function again, as it is a neuron made up of neuron, same, it controls the reflex functions, right. So, if you see the section of the spinal cord, in the section, in brain if you take the outer matter, we call it as grey matter, and inner matter you call it as white matter, whereas here in spinal cord the outer matter you call it as white matter, inner matter you call it as grey matter, just opposite. Okay, just keep in mind, brain outside grey, whereas here it is inside grey, grey matter and white matter. And if you see the section, there are grooves, dorsal and ventral grooves are there, like how we have gyri and sulci, the same way we have grooves here, okay, which is dividing like this. And then the other nerve which is connecting to the other, like motor nerve, other nerves, sensory nerves, relay nerves, etc. So, this is the structure of the spinal cord. You can see the dorsal root ganglia, this part, sensory neuron, receptor, it may be a muscle or any other part sensory. Okay. So, we have completed totally about the central nervous system. First part we saw the brain, and now we have completed the spinal cord, as well as we completed the PNS also, peripheral nervous system. And next, we know the system on the whole, how it is functioning. Okay, brain we know the function, spinal cord we know the function. Who is the carrier? Who is carrying the message? That we need to take. Like if you take your whole system, there is a functional unit in your system. We call that as cell. Like that, if you take the brain and the spinal cord, there is a functional unit called neuron. That neuron plays a major role, and how it is going to conduct the impulse, the generate and propagate, that we are going to see. So, before we are going to see about that function, we will talk about the properties. Any part if you take, you should know about the property. So, if you take a cell, you know the property of cell. Like that, the property of neuron is very important to learn its mechanism. So, this is the structure of neuron. In the first part, we discussed about the structure of neuron. You know, it has a dendrite, axon and then nerve ending, and how the impulse is generating and then propagation, the travel I am talking about. You can see the parts we have discussed already. So, we will move directly into the properties. The first property, excitability. When the neuron will get excited, a very important thing you have to see, if this is a structure of neuron, I am just drawing only the axon. Okay, I am just taking only the axon. If this is the neuron structure, I am just drawing a flow diagram. I am taking only the axon structure. This axon structure as the layer, two layer and inside. This inner part you call it as intracellular. Outer part you call it as extracellular. Okay, and then here it consists of some substance that we are going to see. So, this structure we are going to see in detail in the next slide, how it is going to conduct the impulse. This part is very important, which is extracellular and which is intracellular. And then the layer you call it as plasma membrane in the cell. Here neurilemma. Okay, we call the layer as neurilemma, the outer layer neurilemma. Plasma lemma, like neurilemma we will call it as. And then this structure, extracellular fluid, intracellular will take it. Intracellular fluid, what you will call it as? For example, if it is a cell, inside we call it a cytoplasm. Like that, inside this neuron, that plasm, that jelly-like substance, you call it as neuroplasm. Only thing we are giving the thing like prefix and suffix, that's all. Okay. So, here neuron, since it is neuron, we are going to call it as neuroplasm. There cytoplasm based upon the cell, right. So, here it is neuroplasm. Okay. So, this is the structure of the axon in detail. In next slide still more we will explore this one. So, what is excitability? Excitability when they are going to conduct the impulse, when stimulus in these conditions. Okay, in these conditions you can get a stimulus. I mean the neuron can get the stimulus, electrical, physical chemical or mechanical. You can see physical, maybe heat, cold, and then electrical charges, chemical, acid or base, any type of chemical, mechanical when there is an injury. So in these conditions, the stimulus will start propagating or start initiating, we can say. So that you call it as excitability. Okay. And next one, next property. First property, excitability. Second property, we are going to talk about all or none rule. Okay, what is that all or none rule? For example, you take a pistol. In the pistol, there is a trigger. The trigger, what you have to do, you have to press with the correct force. Then only the bullet will be shot, right? So what happens, sometimes the trigger, you will be keep on pressing like this, but it won't be produce any shot there. You have to give the exact force. The same way the stimulus has to be propagated in such a way that is a voltage minus 50, minus 70, what we are going to learn later. So that voltage, that correct voltage it has to produce. Then only it will propagate the stimulus. It will accept the stimulus and the stimulus will get propagated. So that you call it as all or none rule. Okay, you have to produce the exact stimulus. You can see the word there, threshold stimulus, and you can see the number also there, threshold stimulus number. Okay, so in this, when they get this particular voltage, the stimulus start propagating. So that you call it as threshold stimulus. In a very simple way, think about the pistol and the trigger, right? So you can see about this. The minimum stimulus needed to achieve an action potential. When it reaches action potential, propagation takes place. Later in mechanism, we are going to learn in detail how they are going to propagate. When a stimulus is going to happen here, if this is the place, there is a stimulus, how the stimulus will propagate, will move from one place to another place. That we will see in the mechanism. Here we are learning only about the properties, right? And then it's called the threshold stimulus. Please make a note. It is also another important topic for 1 mark. The threshold stimulus causes the membrane potential to become less negative. Okay, the value. I'll show you the graph also, how the graph raises and it's going to decline. Yes. Next property, resting membrane potential. Okay. Potential difference, membrane potential difference or resting membrane potential. Okay. Here you can see the electrode and how it is moving. Just an example. Imagine taking 2 electrodes and placing one on outside, another one inside. Okay, one electrode you are placing outside, another one you are placing inside. The plasma membrane of the living cell. Okay, that is a membrane that says which is out and which is in. Outside the plasma membrane, extracellular. Inside the plasma membrane, intracellular, right? You would measure an electric potential difference between these two. You can see that there's a difference, potential difference, that voltage difference. That you call it as membrane potential difference. So that membrane potential difference during resting stage, how it will be, during action potential, how it will be, how it is going to generate, all those things we are going to see. So first property, excitability. Second property, all or none rule. Third property, membrane potential difference, which is called as resting membrane potential. Okay. So this electrical potential difference is called membrane potential. Make a note, it's a definition for membrane potential. Resting membrane potential, this I'll tell you what is happening here. We'll just see what is resting membrane potential. It is minus 30 millivolt to minus 90 millivolt. So what happens when there is a stimulus, okay, stimulus in one particular place, that will lead into a ion change. For example, current, when you say current, it is a flow of charge. Okay. The same way when you say propagation of the stimulus is nothing but flow of ions. So that's the difference. Make a note, it is flow of ions. What are the ion? And they have a channel also, that we are going to see. So this is the plasma membrane. Okay, one example I am showing here, sodium potassium pump. There are many channels. Okay, one by one we'll see. Membrane, this is a membrane, plasma membrane, otherwise called as neurolemma. And here inside you can see intracellular, outside extracellular. And what is this acting, opening and closing like a gate. We call this as voltage gate. Okay, voltage gate or the channel we can call it as. So, based upon the voltage, in the beginning I told you threshold potential, resting potential, like that we have voltage. So, according to that voltage the channel will open. So, for example, I will take if this is the plasma membrane, okay, this area, if you see from here, you can see only this line. The same thing if I show like this, you can see the layers like this. Okay. So, why I am showing like this? So, from here if you see, you can see only one channel in the plasma membrane. Okay. I am just showing one channel. What channel I am showing here? Sodium potassium pump channel I am showing here. There are other channels also, okay, one behind the other. Okay, when you see in this view, you cannot see it. When you see like this, you can see all other channels here. So, there are sodium channels separately, potassium channel, calcium channel, chloride channel, as well as sodium potassium channel. So, we will see about the channels: sodium, potassium, calcium, chloride, okay, and there is one more channel called sodium potassium channel, okay, both together. So, this is sodium potassium, it will allow the sodium, it will also allow the potassium. So, that you call it a sodium potassium channel. This is very important when we are talking about the mechanism of the nerve impulse. Here we are learning only about the properties of the nerve, neuron. What are the properties, how it is going to work. So, this one in detail we will see in mechanism. Conductivity, the next property. Conductivity is going to conduct the impulse. So, how it is? There is a high concentration of protein and potassium ion in the plasma or in axon. They are talking about inside, okay, high. You can think why I am drawing positive side inside, because there is a channel called potassium, not channel, ion, we can say potassium ion, and there are positive sodium ion. But when you see the even sodium ion will be there inside. Here you can see the sodium ion also, I am just writing small, and here you can see potassium ion also. But when you see the concentration in outside, it is 10 times more. The sodium ion 10 times more in extracellular. Similarly, potassium ion some 20 to 30 times we can say. 20 to 30 times more inside the plasm. Okay, there is axoplasm we can say, or neuroplasm you can say inside this. That is why when you say a channel, sorry, a membrane, here potassium will be more. Okay, will indicate like this, and here you will indicate sodium. So, these ions, again I am saying impulse propagation is nothing but ion change, okay, the movement of the ions. It is not about the charges. So, be clear about this. Next, inside more negative proteins will be there. There are more protein, we are not going to number and name it. So, more negative proteins are there. So, when you draw a membrane, okay, neuron membrane, you will indicate like this, a simple line diagram, and then outside you will indicate with a positive sign, and inside you will indicate with a negative sign. That is it. Why this is inside, this is outside. I will write now, as we are all beginners now, this is inside and this is outside. Outside of what and inside of what it is? It is of axon. Outside the axon, inside the axon, extracellular, intracellular. So, it is negative here. Why it is negative inside? Even though there are ions, potassium ions, more potassium ions, more than that protein, negative proteins will be there. That is why it is negative. Okay, conductivity, what happened? When there is a stimulus in one area, the ion will change. The ion exchange will take place. Movement of ion will take place. How the movement of ion will take place? With the channel. The plasma membrane, I will show you the previous slide. If you see the plasma membrane, okay, plasma membrane of a neuron, here all it is closed. You would have heard about the hydrophilic and hydrophobic structure in plasma membrane. So, it cannot exchange. Only through this channel, the exchange of those ions will take place. Okay, that is why we are using the name channel or the voltage gate. Okay. We will move further. The last property. Okay, before the last, one more important property we will see, refractory period. Okay, there are two types of refractory period. One is absolute refractory period, another one is relative refractory period. So what is refractory period first of all? The refractory period in neuron occurs after an action potential. As we go into the mechanism, the ion change will take place. Sodium will go inside, potassium will come inside. When the potassium comes, what happened? Again it is positive. The positive sign will be there. Okay, again it has to be replaced by the sodium ion. Okay, that is called repolarization, and again it is going to get back to the same phase. That phase you call it as refractory period. Repolarization, the ion, which ion? Potassium ion only will come. Okay, for example, if this is the stimulus, okay, in this place if there is a stimulus, the ion exchange will take place. Okay, like this. Okay, this we are going to learn in detail, just I will tell you a glimpse of that. And then what happened? It is going to travel. This is position. I will write the position 1, 2, 3, 4. And now what happened? It is going to jump to the next position like this. Okay, like this. So where is the sign? It is in the third position. See the place? We call this as conductivity, conduction of the impulse. So how it is going to take place? Because of the exchange of ion. That was the property we saw last time. And now what happened? Again it is coming back as positive sign in the second position. That is because of potassium ion. Potassium ion which is inside, it is going out and making it as positive. It has to be replaced by the sodium ion. Then only the proper channel will work. For that only this phase is important, refractory period. Okay, the same sodium ion will come back to the extracellular place and it will form a proper thing. Okay. So this one we call it as absolute, the types, absolute refractory, relative refractory. Okay. The graph still more in detail we will see in the mechanism. So what is absolute? It is a period of time during which second action potential absolutely cannot take place. So there is no, okay, no impulse propagation. Okay. No matter how large the stimulus, the threshold stimulus, if you produce a threshold or more than that, that time it won't happen. No extra, no action potential, no stimulus propagation. But in relative what happened? A small amount of millivoltage, okay, below the threshold, even that if you are going to apply, it will keep on add together, okay, and then that time it may produce the impulse propagation. We will read this. Is the interval immediately following after the absolute refractory period. In refractory period there is no propagation, during which initiation of the second action potential is inhibited. It stops, it will not allow, but it's possible to propagate the impulse also. Okay. The last one will complete it, summation, the process by which sequence of stimuli, okay, summing, total, I told just now, adding up, okay, they add up even the small stimulus and that will propagate and that starts the impulse propagation. So next class we will see the mechanism in detail. To receive our online lessons, please press the subscribe button and you will receive the latest updates. Thank you.